Hello, everyone. Welcome back to Heng Education, where we share the knowledge of biology. So today in this video, I will be going over to the inheritance, or let's say about genetic calculation and genetic diagram. So it's about inheritance, right? So let's get started. And before going further, I would like to introduce and I would like to remind all the students who are studying this topic, you should be able to familiar with this six terms, or let's say six definition, because that would be really helpful when doing the calculation or while answering the questions, right? Because these six terms are very basic, and then these is a very fundamental part of the inheritance chapter. So let's start off with dominant or dominant allele. So it's an allele of a genes that is expressed in the heterozygote. So when there is, let's say, one recessive and one dominant, then the character will be expressed for the dominant allele. And on the opposite one, it's recessive. So recessive is allele that is not expressed in the phenotype when a dominant allele of a gene is present. And genotype, let's say very simple. So genotype is genetic makeup, or it's the gene character of an organism, which can show the combinations of alleles, and which I will be further demonstrate down there later. And phenotype. So genotype is gene's character. So phenotype, let's put it in a simple way, that is a physical feature or physical characteristic, or let's say physical traits of an organism that result from its genotype. So of course, they are depending on each other, right? So due to the genotype available, the physical features will appear, right? Something like that. So that would be the phenotype. And the last two definition is homozygous and heterozygous. So homo, right? So homo means same and hetero mean different. So homozygous mean a genotype with the same alleles of the genes. And I give example, which we have capital A, or small a, where the both letters, they are the same. So capital or small, right? But for the heterozygous, is a genotype with different alleles of a gene. So that would be capital A and small a, or it can be any alphabet that you choose to do. So it can be capital B, capital D, small d, something like that. So this would be the overall six definition that we should be very familiar while doing the inheritance chapter. So I will go over one more time. So we got dominant, recessive. So dominant is the trait or the character that will be expressed even in the heterozygous stage. And recessive, that is not expressed in the phenotype when there is a dominant allele present. So it's need to both the alleles to be there, to be together, to be able to express in the phenotype. And genotype, let's put it simple. So genotype means genes, genetic characters. So that is inside our cells in the DNA part. So it will be the genetic makeup of an organism. And for the phenotype is physical feature or physical characteristic that result from its genotype. And last two definitions are homozygous and heterozygous. So homozygous, homo, so it means same. So genotype with the same alleles of the genes, and heterozygous would be different. Hetero mean different, right? So genotype with different alleles of a gene. So this all about six definition, and trust me and believe me, if you know and if you understand and if you get this six terms, genetic calculation will be very easy for you to do it. So let's get started with some example, and I will go over those six terms again one more time to make sure that you get it correctly. So example, so in pea plants, tall plants are dominant to dwarf plants. So the questions already stated that tall plants is dominant to dwarf plants. So meaning that dwarf plants, they are recessive, tall plants, they are dominant. So in genetics or in inheritance calculation, to make it simple, if a character is dominant, we represent with a capital letter. And if it is a recessive character, we use a small letter of that same alphabet to be represented as a recessive. So let's use, let's say capital T because tall plant, right? So for tall plant, if it is dominant, so we will represent it with capital T. And for the dwarf plants, we represent with small t because it have to be the same alphabet that we are representing. So if you use, let's say if you use capital F, then it should be capital F, small f. If it is D, it will be capital D and small d. So dominant character with the capital letter and recessive character for a small letter. So now, since they are alleles, right? So alleles mean the alternative form of a gene. So there will be a like how do I say? There will be a two part of the genes, right? Two parts of the alleles. So when we combine, we will have a total combination of three combinations. So there will be capital T, capital T, capital T, small t. It can be small t, capital T, but we represent the dominant character first. So capital T, small t, and then small t, small t. So those are as a chromosome or those are as a allele. So we can say these are the genotype, right? So these are the genotype because they are inside our cells and we cannot be seen and they are the allele. So this is the first definition, which is genotype. And capital T, since capital T is dominant character, so we will see tall plant. So this tall plant is phenotype, right? Because we can see pea plants, very tall plants and dwarf plants. And small t is a dwarf character. So when we have both capital T and small t, and we already mentioned that a dominant character doesn't need both. Only when there is one available, it can also show the character. So capital T, small t would also be a tall plant. So tall and dwarf, they are the phenotype, right? So those are the phenotypes. So we can use this as a phenotype, or let's just say they are the physical features or they are the physical characteristic, right? So we got dominant, recessive, genotype, and phenotype. So now let's look at here one more time. So we got the same alphabet, capital T, and small t, small t. So those characters, they are known as homozygous. So let's just say we will write here. So that would be homozygous. So homozygous where they have the same thing, right? So it should be the same. So capital T, small t, small t. But for the, okay, let me choose a red color. So here we got capital T, small t. So that would be heterozygous because they have a different, right? They are different. So that is heterozygous. So here we can kind of define all the six definition one more time. So we got dominant, where we will use a capital letter to represent a dominant character or dominant feature. And for recessive, we will use the small letter. So that will be a recessive character. And so genotype, we got capital T, capital T, capital T, small t, small t, small t. So those are the genotype and the physical feature that we can see with our eye. So let's say capital T is tall plant and small t is dwarf plant. So we can see a dwarf plant, we can see a tall plant. So that would be the phenotype. And if they have the same alleles, or let's say capital T, or small t, so that would be homozygous. And if it is capital T, small t, so that would be the heterozygous. So I hope you get the main importance definition of the inheritance, right? So after you understand this, the inheritance part will be very easy. So after we know about the six definitions, so let's move on to the genetic diagram. So here I would go over very detailed step-by-step doing. So we will take the same example with tall plant and the dwarf plant, right? So let's say now we got okay, let's say we got one tall plants, homozygous tall plants, and homozygous recessive plants. Okay, so let's say we are going to cross with uh homozygous tall plant crossing with homozygous dwarf plants. Let's say homozygous dwarf plants. So that would be the scenario that we created. So let's get started with step by step. So we have to start off with the parents phenotype. So parents phenotype would be tall and dwarf, right? So this would be the parents phenotype. So after we know about parents phenotype, we have to write the parents genotype. So we take a scenario of both are homozygous. So that would be capital T crossing with small t, right? So we got homozygous dominant tall and homozygous recessive, right? So capital T, small t, and then we will here have gametes, or let's say sex cells. So here they will give you capital T and capital T, or either you can write two bubbles, or let's say two circles, or one circle is also enough because they are of the same. So we got small t. And after that, we will have to draw a Punnett square. So please make sure you use your ruler to draw a very, nice rectangular box to be able to do the calculation. And let's put the gametes here and here. So the first one, we will get capital T small t, capital T small t, capital T small t, and capital T small t. So those are the offspring that we expect to get by crossing those two type of tall plant and dwarf plant. So now this time, it's not about parents, it will be about the offsprings, right? So we get offspring. Let's start with genotype because it's easy. So genotype, so we have all capital T small t, then I would say they are 100% capital T small t, right? Because genotype, so it will be capital T small t. And then here we got offspring's phenotype. So physical feature would also be 100% tall plants, right? So it's me. We expect to have all the plants produced this way would be 100% tall plants. So this is the overview that we have created. So let's go back one more time. So we have to start off with parents phenotype. So we write the phenotype, so tall dwarf, and then parent genotype will be capital T, and small t. And we got gametes, and then do a Punnett square, and then we write the offspring's genotypes and offspring phenotype. Okay, so now this is the first generation. So now for the next generation, we would like to cross the offspring together. So we would like to cross the offspring together, right? So it will be both tall plants. Okay, so let's go back one more time. So here, so this is the first generation, and this is for second generation. For second generation, again, the starting off with parents' phenotype. So this time they are both tall plants, right? So tall plants crossing with tall plants, and we have parents' genotype. So for genotype, they are heterozygous. So it will be capital T small t crossing with capital T small t. And for the gametes, so this time it will be also capital T with one and small t. And here we also have capital T small t. So these are gametes. And now let's do the Punnett square again. So now we got Punnett square. Okay, oh my one, sorry. Okay, so now we got a Punnett square. So let's go. Okay, so we get capital T small t and capital T small t. So for the first one, we get capital T here, capital T small t, capital T small t, and small t. Oh, now we can see a different variety of the offspring produced. So now let's get started. So we have offspring's genotype. So now, offspring genotype, we got all the combination, right? Capital T, capital T, capital T, small t, and small t, small t. So we got one capital T, and we got two capital T, small t, and we got another one small t, small t. So for capital T, small t, it is 1 out of 4. So that would make 25%, right? For capital T, small t, it is 2/4. So it would be 50%. And similarly, small t, it is 1/4. So it would be 25%. So let's get say 25% capital T, 50% capital T, small t, and another 25% small t. So this is the genotype. So now let's continue to the phenotype. Okay, so we got offspring phenotype. So since both capital T and capital T small t, they give us the character of tall plant. So that would make up 75% tall and 25% dwarf. So simply here we got offspring genotype. We can also say 1:2:1 ratio. And for the offspring phenotype, it would be 3:1 ratio. Okay, so this is the sample example that we get from the tall and dwarf plant. So this is the way how you should be doing the genetic diagrams and genetic calculation very neat and tidy. So it's make it simple and it's make it, clear version to both for you to check again as well, like when after you finish doing the calculation, right? And so this would be my first example of where showing the like two generation, right? So first generation with tall and dwarf, and then we got all tall. But when we cross that all tall plants together, they give you all the ranges of both tall and dwarf, and we can see the probability and ratio. So this would be the very first intro simple example for the complete dominant. So after we know about a one, how do I say, monohybrid character, and after we know about a one generation, but what if we want to know more than one generation, and if we want to know more about the family tree, or if we want to know about the parents and grandsons and granddaughters and like everything, then we have to use a pedigree. So pedigree is a diagram which shows a family tree for an inherited characteristic. So in this case, we are using the polydactyly example. So polydactyly is an inherited condition in which a person develop an extra digit on their hands and feet. So it's mean like they will have more than 10 fingers or more than 10 toes. So that would be a condition. And the statement already said that it is determined by a dominant allele. So we already knew that. So dominant allele and recessive allele. So dominant, it will be extra digit. And if it is normal, then that will be the recessive character, right? And the textbook already mentioned that we will use capital D for polydactyly and small d for normal. So we already knew that. Capital D means it's dominant character. Small d is for small letter is for recessive character. So we have capital D, capital D, capital D, small d, small d, small d, and capital D, they will have extra digit. Capital D, small d, they have extra digit, and small d, it will be the normal number of digits. So those are the information that you should be able to process before going to the questions. And those are the things that you need to be able to very familiar with. So now let's look at a pedigree. So another thing that you need to remember with pedigree is in pedigree, a square represent a male and circle represent a female. And if it is shaded or if it is with different correct color in this case, so the red color here represent me, the character that we are looking is present. So here in this case, polydactyly or like where they have extra digit. If it is a white one or if it is just a plain black one, so that would be a normal thing, or that would be the character that we are looking is not present. Okay, so here we have 20 individuals and they are all family related and it is a family tree. So now, what I would like to do together, so you can also do together with me if you're watching this video, and we would like to identify the genotype. So now we know the phenotype, right? So let's say individual 1, individual 2, individual 3, 5, 8, 12, 16, 17, 18. They have polydactyly, so they have extra digit, and the rest that I haven't mentioned the number, so they have a normal digit. So we know that these are the phenotype. Now, we would like to start off with the genotype. We want to identify the genotype. So coming back to our to my normal processing. So we have capital D, capital D, capital D, small d, and small d, small d, right? So capital D, capital D, capital D, small d, they have extra digits. But for small d, it is normal digit. So by knowing this, which one is most easy one to observe and which most easy one to determine? So that would be small d, normal one, right? Because it only have one phenotype and one genotype. But for the extra, yes, extra digit, but they have two types of genotype. So it would make us difficult to go guess directly. So I would start off with the normal character and I will write down small d in all those normal. Okay, so we got here small d, here again, here small d. So I will write all small d to the normal digits. So now that makes a lot easier for us to determine. So now let's look at this first generation. So person 1 and person 2, yes, they have the condition. So of course, it means they will have capital D for sure because they have the condition. But after you look at individual or which are their offspring, individual 4 and individual 6, they are small d, meaning that they are normal. They have a normal number of digits. So very simple. So one small d has to be coming from the father, and another small d should be from mother. So it's very sure that individual 1 and individual 2, they cannot be homozygous, right? Because if they are homozygous, then all the offspring produced will have extra digits. You get my point, right? So if both individual 1 and individual 2, if they are both homozygous, capital D, then all the offspring they produce will also inherit capital D, and for sure, they all will have the extra number of digits. But now, offspring number 4 and offspring number 6, they are normal. They have normal number of digits, which is small d. So it have to be coming from their parents. So one is from father and another one is from mother. So that's why we can make a decision that parents 1 and parents 2, they are both heterozygous. So they both will have capital D, small d. Okay, that's how we have to process. And then moving on to individual 3. So again, individual 3, since it is red color, meaning that, right, he also have the extra number of digits. So for sure, he will also have capital D. But when we observed individual or the offspring 9, 10, and 11, they are all of homozygous recessive, or they all have normal number of digits. So suring that one small d is from mother and another small d is from father. So individual 3 also become capital D, small d, right? Capital D, small d, like that. So you have to think this way, right? So the same thing for individual 12, 8, 16, 17, 18. So we have to process like that because so let's say individual 17 and 18 and 16, they all will have capital D, small d, capital D small d, and capital D small d, because one of their parents, so let's say their mother here and their father here, they have small d, small d. So for sure, individual 17, 18, and 16, they are heterozygous, but we cannot be sure for individual number 8 and individual number 12. So I will leave it with capital D only, because it can be both capital D or capital D small d. So this is how you need to process, and this is the example that where we know the offspring, and then we have to identify the genotype of the parents, right? So for the previous example about tall plants and dwarf plant, we know the parent's genotype, parent character, and we are identifying, or we are finding the phenotype and genotype of the offspring. But in this case, after we know about the offspring, then we are looking backwards to their parents, right? So this is the pedigree, and this is also another concept that you should be very familiar with it. So again, one more time, a square one represent male, and a circle is represent female. And in your question paper, it will just be black and white. If it is white, that means the character that you are looking is not present. If it is black, or if it is, let's say, grayish, or if it is shaded, then it means a character that we are looking for is present. Okay, so this is about pedigree. So after we know about pedigree, another important thing that we should know is test cross. So what is test cross? So test cross is a genetic cross which we use to determine the genotype of an individual, showing a dominant phenotype by crossing with homozygous recessive for the same trait. So let's talk about our example of tall plants and dwarf plants. So I will write the feature here again. So capital T, capital T, capital T, small t, and small t, small t. So capital T is tall plant. Capital T, small t is tall plant, and small t is dwarf plant, right? And it is very obvious, small t, draft will be only one. We don't need to have doubts on that, right? So if it is draft, it's mean small t, right? But for the tall, which is a dominant, right? Which is a dominant phenotype, if we see a one tall plant, we cannot be sure, or like, let's say we don't even know whether they are homozygous dominant or whether they are heterozygous. So do you get what I mean? So with the tall plants, if you see a one tall pea plant, you cannot just straight away tell whether they are homozygous or heterozygous. But if we see a one dwarf plant, we are 100% sure that it is a homozygous recessive. So now a row of test cross comes in. When we want to know whether they are homozygous tall or whether they are heterozygous tall, right? So then we have to cross with a dwarf character. Okay, so it's me crossing with a homozygous recessive for the same trait. So let's say we know tall, and we cross with small t, and we have another tall, and we cross with small t. So this tall is question marks. We don't know whether they are homozygous or heterozygous. And let's just say the offspring produced. So now we know after breeding them, so offspring produced, they has around 50% tall and 50% dwarf. In this cross, they are all 100% tall. So we can see from the offspring, right? If the offspring produced, let's say 1:1 or 50-50, 50% tall, 50% dwarf, then it will be heterozygous, right? Because when we do the pendant square, so I can show with pendant square here again. So we got a pendant square here, okay? And we do not know, so we only know one recessive, and we are only sure with one T, right? If they are both of capital T, then every offspring produced will be tall plants. But here we got 50% tall and 50% dwarf. So to be dwarf, it should be capital T, if it is to be draft, that should be small t, small t. So one small t is coming from the draft that we already knew, but another t where it is coming from, another small t, it should be coming from the unknown, like whether they are homozygous dominant or heterozygous dominant. So now we can be sure. So that would be small t. So we get capital T, small t, small t, small t, capital T, small t, like that. So here, on the other hand, if it is all tall, right? If it is all tall, we only know one, okay, let me draw one more time. Okay, so here we got small t, and here we only know one capital, and all the offspring produced are tall. So there cannot be small t, right? So there cannot be small t. So that should be capital T. So that would be homozygous dominant. Okay, so this would be homozygous dominant. So that's how a test cross play a role. And it is really important when we want to identify whether a dominant character is homozygous or heterozygous, because that would be really helpful in the agriculture of farming. Let's say if we want only tall plants in our garden, then we only want to cross with homozygous tall to be able to produce all tall offspring, right? And another example, if we want one certain character to be produced in our factory or in our industry, then we would like to have the thing that we can yield maximum, right? That the maximum yield can produce. So that's why a test cross is also another important concept that you cannot miss out. So I'll repeat one more time. So test cross is a cross that used to determine the genotype of an organism, which show a dominant phenotype by crossing with a homozygous recessive for the same trait. So we have to cross with homozygous recessive. Okay, so now we got pedigree, we got test crossed. So moving on to Nate's concept, which is about codominant. So codominant is when the both alleles are expressed in the phenotype. So this would be the another concept from the previous concept that we were doing. So codominant is when the both alleles are expressed in the phenotype. So the previous one that we did about the tall plants and the dwarf plants and also about polydactyline, they are considered as complete dominance, meaning that one character is completely dominant over the other character. So that would be the complete dominant. For this case, that would be codominance, right? So codominance means when the both alleles are expressed in the phenotype. And another very typical example that we would come across is flower colors in the snapdragon. So let's say we have C capital R and C capital W. So this is red flower and this is white flower. So I will just go right straight into this. So when we cross these two plants together, so again, it's similar. We will have to do the Punnett square, and we have CR, CR, and CW, CW. So they all produce CR, CW. So there is no one character that is dominant or that is recessive. So both the character will be expressed. So they produced all pink. Okay, so here this CRCW is another character which both alleles are expressed in the phenotype, right? So both alleles are expressed in the phenotype. So this will be about the pink. So when we cross those two pink together, so again, we will have pink crossing with pink. So we have CRCW, CR, and CW. So again, let's draw a Punnett square again. Okay, so we got Punnett square. So here, CR, CW, CR, CW. So first we will, oh my God, this is not straight, sorry. Okay, so here we got CR, 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 CW, CR, CW, and CW, CW. Oh, again, here we got this one the same, this two the same, and this one is another one. So we again got similar when we cross with two heterozygous top plant. But in this case, one interesting thing is that they have both the same ratio for both genotype and phenotype. So let's say genotype ratio. So we got 25% CR, 50% CW, and 25% CW. So which again is 1:2:1. And also phenotypic ratio. Phenotypic ratio, again, we have 25% red, 50% pink, and 25% white. So again, that is 1:2:1 ratio. Okay, so this is about the one example of the codominance. Okay, so this is about the codominance in flower colors in Snapdragon. So codominance means when the both alleles are expressed in the phenotype. Okay, so I will end my first video here and I will come back to my second video about inheritance, where I will be explaining more about codominance as well as with the sex determination and also the sex-linked diseases or sex-linked conditions. So this is the end of my video one, or my first video about inheritance, and I will come back with a second video explaining about blood type, sex determination, and also about the sex-linked condition. So now we discover about key definitions, or the 6 key terms that you need to be familiar with. And then we talk about the genetic diagram in detailed process and in detail step by step. And we talk about pedigree, we talk about test crossed, and we go over the first a few bit of codominance, which is the flower colors in the Snapdragon. So thank you so much for watching about the inheritance as well as genetic calculations and the genetic diagram, and hope to see you in my next video, which is this continuing video, which is the part 2 of this chapter, which is inheritance. And thank you so much, and hope you enjoy my video, and hope to see you all in next video. Thank you so much. Have a great day. Bye bye.